Eh,
Sí, me la puedo quitar, sí. Espera, quitamos aquí.
Hello, everyone, and welcome to the Biolab Conference. Welcome to those online, and welcome all the students from the Master in Design through New Materials here at ANGAR. So this conference is organized by ANGAR and the Biofriction project, and Elisava Research and the Master in Design through New Materials. So today we'll be having guest lecturers Maria Boto, Juan Crespo, and Laura Benitez. They'll be shining some light on what biodesign, bioart, biohacking, and other concepts that we'll be dealing with today. So we will have the three conferences and then an open space for questions, and also some questions from myself I will like to pose to the lecturers. So today, we start with Maria Boto. She's a researcher at CASC, School of Arts, University College of Ghent. And she's going to talk about the ecology of color, a research journey from nature to materiality. So the floor is yours, Maria. Thank you.
Yeah, first of all, thanks. Thanks to both Lauras for organizing this and inviting me to be part of. So today I'm going to explain you a bit about what I'm doing in Kent at the lab of the Royal Academy of Fine Arts. Myself, I'm a scientist. I'm trained as a scientist, but after my PhD here in pharmacy, in the pharmacy school in the University of Barcelona, I decided that I wanted to do something else, something a bit different, and I had the opportunity to join a bio lab as a lab technician, but basically for working for artists and designers. So while I was there, I met some persons from the Royal Academy of Fine Arts, and they invited me to build up a lab and to create a space with new tools and new methodologies for the artists and designers that they were part of the school. From the beginning of the research process, we thought that the lab should be a space for exploring materials and exploring sustainable practice in art and design. And we wanted also to engage as many students as possible. So the thing that we thought that every student had in common was color. As you can probably know, color has a huge environmental impact. Maybe here in Spain, we are not so aware about it because most of the things that are produced with color are quite far away from us, but it is. So we thought that that's the perfect match to bring something that all the students and most of the artists and designers were considering at a certain point in their practice, and that it could have a big environmental impact. So we built up a small lab. Now it's slowly growing with basic tools for the lab. And we started developing some methodologies and some tools and booklets for the students that had not a scientific background at all to feel comfortable working with this type of materials. Now I will start talking about my research. The research is divided in two parts. Basically, I divided the research in the way that the two ways that we can obtain color from nature. The first part is about pigments. So when I started the research, I didn't know anything about color, so the first thing that I did was to look which alternatives were already there, sustainable alternatives, and why they were not longer on use. So with the help of the Industry Museum of Ghent, we started collecting a lot of plants and minerals, and creating a database by testing the different materials on textile, on paper, extracting the color, dyeing, printing. So it ended up like a big database because there are many colors and mineral that has been used for many years. So you can see here some of the parts of the database where all the samples on fabric and textiles and plastic were included, and also with the right page about how we extracted the color, where we found it, how we applied the color. Also, this database is online, so the list of plants, algae, and bacteria that we have been using, you can find it in the website. And also, in this process, at the beginning of the research, we found out that apart from the colors, the traditional coloring materials that we were using, as they were disappearing, also the color names were disappearing, so we created a kind of website where the only thing that you can do is name colors to create another way of keeping these colors, and this is part of this database. Before moving to the next part, I have to say that all this application of natural colors and extracting pigments from minerals, we did it always with the students of art and design, because I was somehow quite curious about why they were not longer considering this type of materials. And basically, they were coming over and over about the same things. The application was much longer, the color was fading, they were much, much more expensive. So the next step, what we did in the lab, was to look for other type of materials that they were not longer considered, that they have never been considered in arts and design as microorganisms. Because somehow, for example, if you want to dye with madder, that is a plant like it's very used for creating a bright red. Since the moment that you put the seed into the ground until you get it and you apply it on textile mainly, you have to wait for 2 years. So with this idea, we thought about microorganisms can be something that can grow pretty fast. And I already had some experience from the Netherlands, so we started collecting microalgae and bacteria and making a list. First, bacteria, because I already had some experience, but quite soon in the process, we found out that the bacteria had some limitations in the application and in the growing, and it was because there are many regulations, and although there is a lab and everything is under control, we are still an art academy, so there is not a special lab for working with bacteria. So quite soon in the process, we found out the microalgae, and I think since then we have been mainly working with those ones. Microalgae are growing as fast as bacteria. You can find it in many colors, and they allow us to approach colors in different ways. One way was by not only growing the microalgae and extracting the color, but also like growing the microalgae and changing the color while it was still alive. So as you can see in this picture, so you were able to grow the algae in one color, stress the algae with light, and transfer it into another color. And also many of these algae, they have some kind of stationary state, so you can use it while they are dry, and once in the moment that, and applied and painted with it, and in the moment that you put this paper into water, the algae come back to life. So these are some colors that you can get by changing the conditions of the algae. Also, the algae, you can have many algae all around the lab, with different cultures, so it's quite easy to keep them alive. And the process of extracting algae is always quite simple. So you grow them, you filter them, at the end you end up with a kind of mass, a biomass. And from the biomass, you can extract the pigment. Here is also explained how you can extract the pigment, but basically, in most of the algae, there are several pigments involved, and what you have to do is to break the cell and then put the right medium to force the pigment to be dissolved in and then extract it. Here is another algae. that also you can, as you can see in this picture, you have the picture of the dry extract. That is something, it's a step further. So if you want to have the pigment dried, you have to freeze dry it. And at the end, we end up with this kind of palette. Now it's growing a bit. We have more greens. So when we have this with these pigments, we start applying them. First on textile. So what we did was to look back again to what we have done before with natural colors, looking for the right binder, looking for the mordants, and applied on textile, on screen printing. We did a lot of screen printing with different colors. Also, we play with different mediums to have different shades of colors. And as you can see here, even there are a small number of pigments, not so small, but still there is a limited number of pigments. By mixing them, you can have a wide range of colors. Here is like a project that we did with an artist in Valencia. He's making notebooks, and we made the covers for the notebooks with the microalgae. And yeah, these are some pictures of our work that we did for Lille 3000, an exhibition in Lille that is about to start, with a massive calendar that is fading away quite slowly. And also we tried these pigments on digital printing. So we extract the pigments and prepare the medium, and with a plotter that it was quite easy to hack. We adapted and we printed some posters. The previous research were also printed. Also with the pigments of the microalgae, we made a filament for 3D printing. We used several pigments, and we mixed it with polylactic acid from potato, and then we made the filament ourselves. Depending on the pigment, we had some kind of texture, because sometimes if the pigment is not heat resistant, can get burned into the process, and then you have this kind of texture in the filament that is quite interesting. So this is all printed with PLA with microalgae pigments. But as I say at the beginning, in the process of working with structural colors, with plants, something that the students were complaining was the fading process. So if you are using a plant for dyeing or for printing, you expect that in a few months or years your colors will change a bit. But in the case of the microalgae, the changing and the fading is even faster. So at first we were a bit disappointed, but then we found that it could be a positive thing for some applications as packaging. And also, like if you were using not the pigments but by the biomass where several pigments were in, you were able to even play with the colors as a kind of a time marker. This thing that happened in textile and on paper, we were also surprised that also in bioplastic was the same. Like after a few weeks of light, the pigment was fading. So somehow it's quite interesting, because you can think about recycling and reuse, because the numbers too, after a while, will become totally transparent, and then you can dye it again, make new filament, and print again. At the end of the previous research, we had a happy encounter with an institute in Ghent. They were working with different algae for cleaning water, and we sent them our algae to check if these algae were able to clean water, and we found that they were not only, while growing, they were not only producing pigments, but also cleaning wastewater from the industry. So we start thinking about this project, like, okay, maybe we can create a circular system where pigments is a byproduct, but everything that is produced, the loop is totally closed. So since September, we got a new project, and we want to focus on this circular aspect. So now we are collecting wastewater from different industries connected to design, analyzing the wastewater to check if we were able to grow microalgae in there, and also collecting wastewater from our school that normally is quite colorful, and testing them with different microalgae, also testing them with different concentrations of wastewater, and lately testing them on inkjet printing, because something that also we had in mind is like if we can if we can put this printer in an inkjet printer, like a home printer that works with these microalgae pigments, and you can use it for printing paper or other products with a short life, it will be perfect. And now we are in this process, measuring the particle samples, fixing the viscosity. And now I'm going to talk a bit about the second part of the research. This started in September with my colleague Hélène Saint-Aubin. Already a couple of years ago, I had the opportunity to go to a conference, and in one of the speakers, Matthew Soke, say like he will be able to create any colors and not pigment dependent. So I was quite fascinated with it, and then I got in contact with Matthew, and he put me in contact with this network of scientists who are working on structural coloration. For those who don't know what structural coloration is, it's basically color that is not because of a pigment, but because of a nanostructure. So there are some nanostructures that with the size of the visual spectrum that can, by effects of diffraction and reflection, can get different colors. So here you can see a picture of Liliana. How, in the case of birds, only the colors that you have in the upper part are because of pigments, and the colors that are in the down part are because of structural coloration. Also, many insects are the colors that you can have there is structural coloration, and of course, birds, hummingbirds. These structural colors, they have also iridescence, and also sometimes these effects like these super blacks. How this structural coloration works, here is an example on birds. So as you can see here, there is only one pigment, that is melanin, and it's between keratin layers. Because how the light travels through the melanin and the keratin, depending on the angle, you see one color or other. Here is another example in a duck feather. So when I'm talking about melanin and structures of melanin, we have to go to nano, so to the last part of the presentation, of the slide. And these structural colors are also responsible of butterflies, and these are also more butterflies. I think probably this one is the one that is the best known. So you can see this nanostructure that creates this blue. So this, although you see blue, I don't know if you ever touch a butterfly, but if you touch it, everything is black. So you are just taking these black scales. And also in nature you also can find structural colors. In this case, it's because of the cellulose. Cellulose also has the capacity of create crystals, and if the crystals are organized on the right way, you can have these iridescence effects. So since September, we have been working in this project using eumelanin, so the black melanin. There are different types of melanin, but we have been working only with one, with the brown melanin, having synthetic melanin in the lab, and then trying to create this structure. Here you can see this is a layer, a film made out of melanin, so it's a brown color. But if you move it, depending on the angle, you see how the color appears. So it's quite magic. Here is also another layer of melanin. So you can have, as my colleague Matthew say, basically any color, and you can have different effects. So here more pictures of melanin. You can have it matte, metallic. And now we are learning how to create the right conditions to control somehow the color that we are obtaining, and to make these structures a bit more stable to manipulate and to transfer. So here you have more colors, greenish. Here there is something. If you compare these pictures with the first one that I showed you, where you see just the brown color. Basically, what we are doing here is that we are leaving a background of melanin at the bottom, so that increases the contrast, and that's why you don't need to play with the sample to see the color. You can have a crazy amount of colors, and also the movement makes the colors look different. We have been analyzing the colors, and in any sample there are many colors, and I don't know if you can see it here well, but many of the samples, when we analyze it with a spectrometer, we see two peaks. That it means that they have iridescence. And in the same, what we see is like a flat layer that is what is in contact with the air, and then below the balls of melanin organized. We have been trying to translate this basic research into an application. We have been working on textile, trying to create these films on textile, first putting a layer of melanin to generate the contrast, and then putting the nanostructure on top. Also on flexible plastico, flexible thermoplastic or something like that. Also, in this case, I think we already used like a black plastic to make sure that the contrast was high, so we didn't have to add more melanin. And also because my colleague Helen is a big fan of clay and ceramics, we have been playing with it. And that's something that it was quite interesting, because as I saw you in one of the slides before, in nature you never find pure melanin, but you find melanin with keratin, air, chitin, many different types of materials. And we have been mainly using melanin, but while working with ceramics, we were playing with glazes, and that's something that it also affects the way that we saw the color. So depending on the type of glazing that we were using on the porcelain or on the clay, you can see how the colors can become more metallic or matte. And also, yeah, you can go bigger. This is also clay, but with no glazing, so the color was totally matte. And also, in the process of this structural coloration research, we met a Belgian artist, Veronica Johnson, and she was interested in research the sensorial potential of structural coloration. So this is some of the works of Veronica. And with her and with the PSL and L'Oreal, we are working on how to create something like to create like a kind of a color experience in a room. And I think that's it. I don't know if I'm on time or... Yes, I had many slides. Okay, thank you. You're welcome.
Hi, thank you, Maria. So we have now Juan, and thank you for this exciting. I'm amazed on the structural colors. We'll have some questions for you after Juan and Laura. So Juan Crespo, he's done his PhD in biomedicine, biofabrication, etc., and he will teach us on his path. And he's responsible of the Design and Living Systems lab, Biolab at Elisava. And today he's going to talk about growing engineering and design, and thus bio. So the floor is yours, Juan. Thank you.
Thank you very much, Laura, for your presentation. And thank you, everyone, for inviting me and for coming. I would like to start my presentation by looking to... I found this post like a couple of days ago, and I thought, well, it says, in the entire universe, wood is certainly more rare and valuable than diamonds. And I thought, that's an interesting point of view, because here in our planet, in Earth, some people, they seem not to value what it grows, let's say, like the living things. In fact, we find more valuable diamonds and gold than nature itself. So I think this is going to be like the very key point and the key aspect in my presentation. And now, I'll jump to the proper presentation. Growing materials. Just if someone is asking, what is this on this first image? Some of you, I know, you know, because I explained it to you in class. But for the rest of you, yeah, I might be repeating myself a bit. Never mind, I'll try to make it like more. This is the ass of a spider. That means when they excrete this silk, this spider silk, they can choose the diameter of this silk, the consistency, and the amount of different wires they're going to be like pulling out from there. So it's very, let's say, nature's engineering is far beyond of what we can imagine so far. So Ron Arad said there are 5 processes available to the product designer. Wasting, which is what we mostly do in this planet, extracting things from nature. Molding, that would mean conferring a shape by pressure to a material. Forming would be adding parts, but not as in an assemble point of view. And growing. This last point, growing, is the one I think is more optimistic regarding the future. Why? Because nature only grows materials. That means we can only find growing things in nature. There's probably a debate around if a rock is alive or not. I'm going to leave it here. We're not going to get into this debate. I know some of your opinions. Look at what we do, we humans, not all of us, but a good part of us. A tree. Designers, and mostly engineers and many other people, we are used to, well, we get a tree, and we obtain a material from this tree. That is, we extract. We don't assemble, we don't conform, we don't mold, and we don't grow it for certain. We just cut the tree and we build a chair. This happens with cotton. We can grow cotton and then we fabric. We make pieces of clothes, clothing, garments, whatever. It doesn't change reality, and reality is just the way it works. Nature does it. And then we find these magnificent kimonos or whatever, or shirts, and some other types of garment here. So never mind. I'm just giving you some examples of how we are used to extract from nature rather than working with her. The spider, you know, I love this. And from an engineering point of view, it has amazing properties. Bees, which they actually puke their houses or their housings. They get these almost fractal ways of building their houses. Termites, which are really interesting because they build just by mixing saliva they produce, another sort of puke, and they get to build. Character in this presentation would be bacteria. Bacteria are so amazing, and we use them, of course. We have, you know, we are all covered in bacteria and fungus, but we are not aware of that. But we just have bacteria in all of our surfaces, and especially in the mucosa, because in this wetness, they live and they cohabit with us. But for example, we fabric or we make yogurt thanks to this bacteria. Our digestive system works because of this, the existence of this bacteria. I found this report which is pretty interesting. Like there are some bacteria which are actually fabricating other cells certain polymers. Polymers, not PLA or PLGA, but you know, certain technical polymers that so far we've been synthesizing in the laboratories. Now, let's put things forward. What I would like to introduce is the concept of being a product shepherd. That means instead of extracting materials from nature, can we guide nature to produce our products? We have many examples of this, but not in the way I'm imagining, but we use bushes for making these arcs and these magnificent gardens. But there are also examples where a human being has like twisted nature just to conform this or make these structures. They are not useful, they are just decorative, but we can use them for making something more useful, such as chairs, for example. What if instead of going to, for example, IKEA and buying a chair for 20 euros, we could actually grow our own chair? That would give us so much time to think, deep think, on whether we really need this chair or not. I mean, if it took like 4 or 5 years to grow our own chair, would that be, I mean, we will be growing like 10 chairs for our house, or we just will be growing like a couple of them because we really need that chair. So I think by growing our things or trying to grow our things, we will get to a point in which we will do some reflection on if we really need what we are buying or consuming. There are several examples which are pretty fun. Now, in Elisava, we are working especially with two, let's call them natural agents, such as mycelium, that you may have explored, which are, mycelium is basically a part of a fungus in which the structure is pretty much the same, just hyphas, they just grow. And they are used, this like would be, the mycelium would be called like all the roots from this fungus, and the actual mushroom would be the reproductive part. Of course, it's a fractal, and they are used, they have been used for making some packaging. In some subjects on design, not engineering, in design we have used them too, to grow things. And I wanted to test them in the laboratory with you guys, well, with you guys of the master, but we didn't have time to do that. And we didn't get to do it the proper way, but still, the experiment, I find it like pretty amazing. There are also some designers that especially work by growing these products. I insist, the basic idea here is growing things rather than cutting, extracting, or transforming things. So there are many examples of them. Here are some few pictures. It is also used in architecture. That's why like they've done some mechanical testing to these mycelium-made bricks, and they found that the mechanical properties are pretty astonishing. And another material you may know, it's bacterial cellulose. There are many examples too, in which the bacterial cellulose is actually like an extracellular matrix bacteria make, like an excretion from their, let's call bodies. And it looks like this. It's very like, artistically, I think it has a high amount of color expression. And this is Suzanne Lee, which she was pretty famous by publishing an entire jacket made of this material. And the basic idea surrounding it, I think, is the same. Instead of extracting or cutting a plant for fabricating or making this jacket, she just grew it from bacteria. At school in Elisava, we had last year a very interesting project called Monoma by two students. They also made like an investigation on how to use and apply these new fabrics to fashion. And it's also been used, and we've done this in another subject of design grade in Elisava. It's used for packaging. So again, rather than using synthesized or artificially synthesized polymers in a laboratory or in a fabric, we just can grow them, which would be much more regenerative from environmental point of view. And I think now it's the key for going through a sustainable future. So let's rethink growing. Well, there are several examples too about like Neri Oxman, which made this geodesic cupole made of silk, worm silk. I love this example particularly. It's a concrete which is mixed with a certain type of bacteria. And this bacteria has the property that it reacts when it finds a certain amount of oxygen or different gases. So when a crack happens in a piece of concrete, bacteria reacts and gets to fill this crack. So we can call it a self-healing concrete. Pretty interesting. There are also several companies that make artificial leather. It's actually fruit leather from apple skins, pineapple skins, and so. That backpack is an example of what they make. Again, they're producing it. These are some pictures from last year's subject, in which I was the tutor, and we made like fruit leather, let's call it like that, for packaging. You know these examples. This is made about sand, pure sand, mixing with some glutinant, like so it's like natural types of gelatins. And regarding this growing thing, let's jump into my actual specialty, would be tissue engineering. I did my PhD on tissue engineering, and now currently we are working with the International University of Catalonia, UIC, and we are trying to explore different ways of making scaffolds. Scaffoldings, for those of you who may not be familiar with the term, it's fabricating the temporal housing for cells. That means if we want to heal or restore a function for a tissue that's been damaged for any reason, we need to get this temporal housing for the cells to grow and mature in this housing and then be sequentially substituted by the host tissue. So it's a temporal housing for a cell, basically. This is the definition that you may find in Wikipedia. It's a biomedical engineering discipline that uses a combination of cells, engineering, material methods, and suitable biochemical and psychochemical, physicochemical, not psychochemical, sorry, factors to restore, maintain, improve, or replace different types of tissues. So there are 4 main characters in this equation, which would be scaffolds, the house, cells, like the actors, the actual individual that manipulates the scaffold, signals, which are basically biochemical signals for growth factors, drive cell functions, different things, and bioreactors. Bioreactor would be considering the whole system as one thing, like the interaction of all these people working together. So the basic scheme would be what I did in the laboratory and what I'm intending to do in the new laboratory in Elisava, I'm going to explain it later on, would be obtaining a biopsy from a patient, isolate the cells, expand this culture, fabricating separately from this scaffolding, mixing these cells with this scaffold and these growth factors, and then implant this new thing into the patient. That would be if you may suffer from a heart failure, well, not heart failure, just a little heart failure, a heart stroke. We could obtain cells from your heart, expand them, seed them on this scaffold, and replace your dead tissue from the heart, your dead myocardium. This is actually not happening so far, but I think it's going to be happening in 10 years' time. So in this context, this bioprinting regeneration project has been born. We are working with as I said, with the International University of Catalonia, which is this. They are called themselves the Bioengineering Institute of Technology, and they actually work with scaffoldings, but just for bone tissue. They are focused and centered on bone tissue. So the idea is the same. We're going to use cells, we're going to use a scaffold and growth factors for creating this artificial bone, grown and made in a laboratory, and then try to implant it in the patient, whatever. We don't know the specific pathology of the patient still. So the part of the scaffolding is the thing we're going to develop in Elisava, as we are pretty experts on 3D printing and designing, you know that. So just for those of you who may not be familiar with the term bone, this is a very short class of histology and cytology. This would be a piece of bone. It has 2 very differentiated parts, cortical part made of osteons and the trabecular part. So these 2 combination, every bone has these 2 parts. The inner part, which is called the medulla sometimes, is this trabecular, which is very porous. And the more solid thing is the cortical. So you can see this osteons is the compact part of the bone, and it's made about many fibers. And these fibers are made with collagen fibrils. Yes, collagen. That is some sort of gelatin. And several depositions of hydroxyapatite are deposited in the extremes of these collagen fibrils. And you can go, you can see here a scale. So the idea would be fabricating this scaffold, put the cells inside with some growth factors too, to mature them in the laboratory, and then inject, graft, or just implant this construction into the bone failure. In this real project, what we're going to do is we're going to use CAD design for designing the specific scaffold we want to build. We will be developing different hackings of these 3D printers using different nozzles and designing different nozzles like this, for we need to increase the specific surface area. I will explain what is that. Creating more or less a 1 centimeter cubic piece of tissue and then implant it. That's our main goal in this project, together with the Bioengineering Institute of Technology of the Week, of course. So the first thing we are doing, and this began like 3 months ago, 2 or 3 months ago, we need to increase the specific surface area for those of you who may not know what this term means, it's increasing the surface for the same amount of mass. That is, if we have 1 gram of material, if we use this shape, this star-shaped nozzle, we will have a much bigger surface than if we use a circular surface. I hope this is understood because it's main. An example would be using this manga pastelera. I'm sorry, I don't know the term in English. This thing for, when you do this sort of cakes. So you're increasing the very surface by the same amount of material you're using. So in this context also, it just has been born, the new Biolab in Elisava. This is like in the first floor. This is the very end of it. And we have 2 printers here, microscope and very little more so far. It will be growing up. And these are 2 clone printers. This first printer is like a normal FDM printer. That means it extrudes a melted polymer. And the second one, to be this one on the left, we have hacked a bit, and we have changed this extruder nozzle for a paste nozzle. That means we're using syringes instead of melting or a nozzle for melt material for creating these scaffolds. And these syringes, we can fill them with different hydrogels, for example, or different materials. And in this way, we can control porosity of a bone. This is a good example of how, if the porosity increases, this would be when you have osteoporosis, the pathology, it's because this bone decalcificates and it creates very big pores in the inside, so it becomes very fragile and it breaks. So by using these 3D printers with both systems, I guess we will be able to control this porosity, both with hydrogels and polymers, and try to make really effective scaffoldings for regenerating bone tissue. So we are now doing some, as you can see, different designs of these scaffoldings and using different syringes and hydrogel mixtures. We are also designing, these nozzles are really, really small, and they are not for sale because they don't sell them. So we are fabricating by ourselves with other 3D printers. It's funny because it's a machine that's printing a machine. So I know it's a funny concept. And this is like 0.8 millimeters, and we are trying to increase here the specific surface, as you can see. Some of them didn't work, but some of them have worked. And we are trying with different materials, different hydrogels. As you can imagine, a hydrogel, it would be like a gelatin. It does not have the consistency to bear loads by itself because it just melts. So we're trying to use different amounts or proportion rates of these gelatins or agar-agar and different polymers. And here we are so far. So that would be it. I think I'm on time. Thank you. That would be it.
So thanks, Juan. So last but not least, Laura Benitez. She did her PhD in philosophy, and she's expert in being able to connect philosophy, art, and technoscience. And today she's here mainly as partner leader, project leader for the project Biofriction, which she will present and share already the first results, I guess. So thank you, Laura. The floor is yours.
Well, okay. Thank you very much to everyone, to the organizers for inviting me and making this possible. And of course to Elisava and also Angar for hosting, as always. And yeah, in terms of giving thanks to Angar, I also want to mention thanks so much to Ludovica, to Marc and Matteo, who are the kind of infrastructure, the condition of possibility to be here today together and having also streaming. So I'm going to share my presentation. And of course, sorry, thanks so much to Juan and Maria for their wonderful presentations. Let's say that my presentation is going to be, okay, a bit different in a way. I mean, is not going to be, or I'm not going to go so in deep, to say so, in terms of technical stuff, and of course it's not my role here today for sure, but I will try to explain you how are we trying to approach experimental practices with biomaterials within this framework that it's called Biofriction, and I'm going to tell you a little bit about how I'm putting in this title, Bio Disruptions on Scale, Cares, and Interdependence. So, as Laura said, I have the honor of being the director of Biofriction. Biofriction is a Creative Europe project that is led by Angar in collaboration with BioArt Society, based in Finland, Kersnikova, based in Slovenia, and Cultivamos Cultura, based in Portugal. And as a project, it's let's say, a small project, small in European terms of scale, which is quite big. Is a project of mainly transnational mobility, so you can imagine how we are dealing with transnational mobility within the framework of pandemic, but we are managing to host the artists, so not so bad, at least. But the idea of Biofriction is, let's say, to work within bioart, the so-called bioart practices and biohacking practices. And for example, the title of the project, it refers precisely to this, let's say, combination of biotech, biology, fiction and artistic practices as a surfaces of friction. And here I would like to remark that mainly fiction within biofriction is addressed in a pretty, let's say, different way that sometimes is addressed in general terms in design practices, because fiction here is not just understood as a, let's say, as a critical potential in order to, for example, imagine or design or articulate possible futures or speculative projects, but also, of course, that it's present in biofriction, but also we are trying to deal with fiction, let's say, as a problematic also tool in, for example, when we refer to fiction, we are also like thinking in, let's say, the power of a science fiction, but also for us it's important to refer to fiction as, for example, the perverse fictions and their hyper-real consequences. And for us, for example, it's important to keep in mind, for example, let's say, one of the most obvious perverse fictions that are operating nowadays is, for example, frontiers, like borders. And it's a fiction, but it has like hyper-real consequences, biopolitical ones, necropolitical ones. People is being killed every day because of this perverse fiction of borders. So within the general framework of biofriction, we are working with this, let's say, at least two levels of the notion of fiction. So, as I said, for us, it's pretty important to work within an ethical and a political approach to this possibility of experimenting with biomaterials. And I know that there is a huge debate on what we could call biomaterials or no, but I'm using biomaterials here in as a general term. And also, for example, maybe philosophers such as Eugene Thacker would say biomedia. It could work also for us, but in any way, for us, it's important to how to, let's say, to approach in a critical way this experimentation with biomaterials. What is happening there? What kind of questions are emerging there from these interactions within these sometimes beings, sometimes semi-living entities, and also, again, it appears this debate in between the, a dualistic conception of living and non-living. And precisely, you know it pretty well, the experimentation with, let's say, these biomaterials are showing us that there are super different scales and multiplicity of scales, and this kind of dualistic structure is not so clear, or at least it's becoming problematic to sustain a dualistic structure or conception in between the living and the non-living. So, in our case, for example, here you have some of the projects that are the artists in residence within Biofriction. I'm not going to explain the projects, but just I'm putting this here because I invite you to check the website and to check the projects, because there you'll find the descriptions, the links, and some videos, also some recordings, some materials. But let's say that in general terms, for us, when I was saying that Biofriction is this kind of combination in between these conceptions. For example, also, I think that it's important to remark that within Biofriction, biology is not just understood as, let's say, life science, is also understood as a discourse. So for us also it's important to see and to pay attention to what kind of critical tools we can articulate together, because it's, like from this conception of DIY, like do it yourself, or to do it together. We are more in this do it together, because we are pretty convinced that we are never learning alone. We are always using materials, also, for example, in historical terms, in material terms, so we are never alone generating knowledge. So the conception of the project, having this note of working or experimenting with biomaterial or biomedia, is, let's say, a transversal conception in terms that biofriction is based, or it has the, let's say, the proposal to address knowledge beyond a disciplinary conception of knowledge. So that means that for us it's pretty important to work within biomaterials and biomedia from a situated perspective. And here you have kind of a statement how we are trying to work together, to learn together, to produce knowledge together. And for us, we are not working within a conception of universal rules, methods, or methodologies. We are pretty convinced that every project, it has its own methods, and of course can be rearticulated and even de-articulated. So for us it's pretty important to bear in mind these, let's say, conceptions. For example, we are not so much focused on what is known. We are not so much into, for example, a kind of a genealogy of what does it mean when we are using the term biomaterials or biomedia. We are not so much into this what is known, but how it is known. For us, processes are extremely important. The ethical commitment, the political commitment, and of course I come from philosophy, so I'm going to say philotechnicism, but the ontological conceptions or articulation, and also the epistemological ones. No? For example, this slide here, it presents a typical diagram or scheme, if you want to say so, of situated epistemologies. So for us, it's important to bear in mind what is been written here. Like we have always these situated knower, situated knowledge, and epistemic conditions. So in every practice within biofriction, we know and we try to care of these complex, interdependent social nature culture systems. And for example, this is related to what Juan was mentioning before, because also for us is important, or at least biofriction was conceived within this nature culture continuum. So we are not working from a conception of, like a dualistic conception, culture and nature. For us, there's no separation in between culture and nature. So, and of course, I'm referring to, for example, the theoretical and not so, or not only theoretical proposal of Donna Haraway, but because, for example, precisely for us, this split conception of nature and culture is or at least one of the consequences of this conception is what Juan was explaining before, no? This conception of extractivism, no? This political extractivism, like it seems that this kind of, let's say, abstract entity that we've called nature for so many centuries, it seems that it's only a kind of place in order to extract resources. And we are not working here within biofriction with that conception. So also for us, it's important to bear in mind a situated knowledge, what means it refers to this critical and cultural analysis of, for example, we are always trying to pay attention to the material conditions of these projects. Not just who is, for example, the artist developing these projects, but, for example, who is participating, but also who is missing. No? Not only working on presences or presence, but absence, for example. No? Which are the material conditions, the resources, where they come from. For example, in this case, it's pretty obvious because it's funded by the European Commission. But for example, for us, it's also important to think of let's say, what is going on within the processes of these project developments. No? For example, sometimes what happens with the so-called bioart practices, not so much with the biohacking ones practices, well, it depends on what kind of biohacking we are referring to, but sometimes, for example, within these bioart practices that are extremely catchy in visual terms, they are generating an extreme waste. No? We are, for example, doing something that is super catchy in visual terms, and it seems that it's even interesting in conceptual terms, but which are the levels of waste that we are producing during the project? Also for us, these questions are important, no? And is this let's say, at least intention to take care of of a situated analysis and perspective, and also, of course, these communities based knowledges, no? As I said before, who have the access to participate, who is missing, what is happening there, what kind of relationships, what kind of power relationships also are happening there. So all these things for us as a project are important. And here you have a poster, because also I think that it's important to say that Juan was referring to this wetness, and wetness for us within biofriction is pretty important. And for example, as I said, biofriction is led by Angar, so let's say that center of operations of biofriction here in Barcelona is the wet lab, the place where that you are, like cohabiting during this week. And it's not a biolab, and the fact that we are not using the term biolab, it's also a political statement in a way. So for us, this wetness is extremely important, because within biofriction, I mean, we are, let's say, we are not working, or we are not departing from this conception of an aseptic biolab. The project is based in a trans-hack feminist perspective. That means to work together, hacking with care, and this hacking with care is related to the fact that we are trying to articulate, let's say, active resistances to, for example, some hegemonic narrative and its consequences. Like, as I said before, for example, as we are trying to pay attention to how some perverse fictions have hyper-real consequences, the same happens with, let's say, hegemonic narrative on gender, on subjectivity, on, for example, some power relations as well. So this is the core of biofriction and comes from the Trans-Hack Feminist manifesto that also you can check online. So for us it's important, this kind of commitment to hacking with care, and not, not using care or cares as a kind of a hype, like a cultural hype, because, like right now is a term that is being so much, or so it's so used that it's been even abused, because sometimes we are depoliticizing the term within the kind of practices that we are, let's say, producing. But also, for example, for us, it's important, the second point, to try at least to decolonize technologies. And this is even is difficult and sometimes is super problematic. For example, right now, me explaining biofriction and mentioning how we need to decolonize technologies, and I'm doing this kind of contradiction of like articulating epistemic violence in a way. So we are also full of contradictions, and we are holding contradictions. And for sure, for us, is extremely important this conception of working within queer, feminist, and anti-racist and anti-speciesist methods and pedagogies as well. And precisely for this, I was remarking the fact that wetness is so important for us, because, for example, some of the practices you were mentioning, bacteria, we've been doing yogurt with vaginal fluid, for example, this kind of wetness that can happen in a space like the wet lab, and in this poster that was designed by Belén Soto, who used to be the coordinator of the wet lab some time ago, it says that it's a wet lab, is a lab of fluids, is a sticky lab, is a, well, you can, maybe I can send you a translation. So it's not the aseptic conception of a biolab. So, and here, for example, for us, it's also important, and I think that it's important to think on, contemporary ethical questions and contemporary political questions that emerge from these experimental practices. And in this way, for us, as I said, is also to think of the possibility of more meaningful alliances, and alliances that are not just referring to human agents, and I think that this kind of practices, and also the kind of practices that you are developing, experimenting with different biomaterials that in a way are also bioagents, can, like articulate more potential alliances that at the same time are opening up new or re-articulation of previous questions in terms of ethics or in terms of politics in a way, no? So for us it's also, and here we have this water beer, it's important to think and to work within this experimentation with biomaterials, with this kind of estrangement, no? This experimentation of multifocal scales that are not just the human ones, and what is happening there. For example, what is happening when we are not just dealing with the visual regime, and some other experiences also can occur, for example, with sound, with different frequencies. What happens with when we can experiment, for example, very low frequencies, or what we've been calling with an extremely great group biosonic agencies, for example, what is happening there, or what happens when, for example, you can become a kind of an stranger through your own voice, because you are also experimenting with these biosonic agencies, and here you will find some information on the project of Adriana Nouv, working with this experimentation with voice and biosonic, bioagents. So, I mean, it's important for us to search for commonality, but through difference, and that is, I would say that that's a kind of important point within biofriction, or even we could say, and I'm using the conditional because I'm pretty aware about all the inherent problems within this kind of a statement, but I could say that for us, we are trying to de-articulate, or at least trying to remark the problems of hierarchical conceptions in terms of human, non-human, in terms of gender, in terms of scale, and all the possibilities that we can work on it from there. So, of course, we are pretty aware that even within biofriction, the access is, let's say, is not open to everyone, because, as you know, we don't have a global world. We have a global capital, so that's a thing. But for us, it's important to think within these biomaterials and bioexperimentations on, let's say, different configurations of otherness, and also different conceptions of kind of an extended subjectivity, an extended self, and if you want to say so, a non-ecstatic identity, for example. So as Maria and Juan were referring before to, this experimentation with biomaterials also, in a way, hack the human-centered conception of life, of the world, and even of, let's say, this kind of center conception of the subject, no? Because we are all a multi-community, a multiplicity of bacteria, of different bioagents that can disrupt this, let's say, hegemonic systems in terms of, well, the inheritance, Western conception of subjectivity, for example. So for us, in this sense, it's important to bear this in mind. And for example, what happened, this is a concept by Donna Haraway, worlding, how one of the central questions for us in biofriction is how we are, like being together in this world. And again, this being together, it doesn't refer just to the human subjects or human agents. Even is not just referring to the living ones, because, and we are pretty aware of the controversial debate about what is life, what does it mean living and non-living, but let's say different entities. So for us, for example, as I said, it's extremely important the kind of ethical and political decisions that we take when we are articulating a proposal. And for example, what does it mean to reject to work with bovine serum, that it's a super normal, let's say, biomaterial present in this kind of techno-scientific extended body, that it's a multiplicity of biolabs all around the world. And for example, to artists right now, Vanessa Lorenzo, by the way, who is a former student of Elisava and one of the artists in residence in Biofriction, together with Teresa Schubert, another artist. They are trying to develop, instead of using a bovine serum, because for them is an ethical problem that affects their project, they are trying to develop and to, let's say, to experiment with menstrual blood in order to substitute this bovine serum for a different one, that it hasn't this conception of extractivism from the non-human animals, for example, or some other different practices. So, in general terms, I know that is a more, let's say, abstract or conceptual approach to how to experiment with biopractices, but in this case, for us, it's important what Deleuze was quoting here, that a body can be anything. It can be an animal, it can be a sound body, it can be a mind or an idea, it can be a linguistic corpus, it can be a social body or a collectivity. And related to that, I just want to read a question of this book that I really recommend you, the Care Manifesto, The Politics of Interdependence. And this is a question that is also present in Biofriction and that I'm going to open as the end of my, let's say, presentation. And I invite you to have this question in mind within your workshop, within this week, and to think about it also beyond the workshop and within your practices as designers experimenting with biomaterials. How can we create a more caring world, one capable of sustainable and nourishing all forms of life? And that's all. Thank you so much.
Thank you, Laura. I was impressed and kind of taking notes of all your thoughts too, and starting to think that this is more complex than I was thinking of before we started this lecture. So I think there's a lot to debate. And I would like to open the floor both online, that you can ask questions through the chat, or hear any of you that would like to ask any questions to Juan or Maria. We are open for questions from you. Okay? No questions yet? Anyone? Everything is clear. Questions, when we say questions, we see any kind of comments or excitement you might feel for some of the things that have been shown today. So, in any case, I have some questions for sure to the speakers. But first of all, kind of the first conclusion I take from all your talks is that Biolab, biodesign, or art, design, biology, all these things connected, it's really linked to the idea of the meaning of life, to the complexity of life and to the fact that we're living in contradictions, right? So I think what I thought was a subject that was more framed into some kind of more structural things, and such as design can do, it's opening up and making us think more than we thought at the beginning. So I think that's a good step forward for designers to think of, to understand the systemic view that biology is bringing into design. And one thing that I was, and I don't know if you want to have a seat maybe, and yes, you three. And yes, so the question goes now. So this idea of the idea of time, okay? I think each of you has touched on the idea of time. Maria, you were talking about fading and then rethinking what color can mean. Juan, you were talking about chairs that take time to grow, and then that makes us rethink or evaluate what we need in our lives. And also Laura, and the idea of situated knowledge, of experiencing and of doing things. I think that brings us into a new paradigm of what time means. So just wanted to reflect on what you think about time and how this might change our perspective of linear living and non-complexity. So if any of you wants to make a comment on this.
Well, I would say immediateness, if that word exists, inmediatez, it's probably a disease, because it makes us not to think on what we really want to do, or think, or whatever. So that's my contribution.
Okay, so
regarding purchasing something.
Yeah, in my case, I think it's kind of funny that since always art and design has been trying to copy what is going on in nature, but yeah, moving out the timing part and the fading and the dying part of nature and the disappearance. So we want what is in nature, and we want to keep it as it is forever.
Yeah, well, in my case, it's risky to ask me this question.
Because of time?
Yeah, because of time, but also because we've been, discussing quite a lot on how this kind of experimentation with biomaterials also are hacking temporality. So I think that there is an extremely interesting possibility of also, bringing some, let's say, we could say in conditional, bringing to the table in order to debate and to think together on it, non-human temporalities. So that's the thing with the scales, and I think that it's super interesting because, for example, a linear conception of time, it seems that is pretty difficult to work anymore beyond having in mind that, of course, is a construct. So.
So it's here when it comes also the idea of regenerative design, and also the idea of, for more market-oriented things, is designing the ephemeral, right? So Maria, this question goes to you. If you're developing colors that might be fading, and so how the packaging industry is going to react to that and to rethink how they do products. Can you develop of that, or what can be, or if you had any reaction from the sector or the ink sector on that?
Yes, I mean, for myself, at first it was also a disappointment when I found out that a lot of all the work that I was doing in the lab was disappearing in a matter of months. But I have to say that rethinking about the potential of this time frame and the potential of the use of color, not only for the aesthetical part, but also as a kind of biomarker, and the potential of applying it in different mediums where time could give you an information, not only the color, but also the time where the color will be. It was a positive thing. And since then, we have been going in this direction, thinking about the recyclability, because a lot of color waste is not, I mean, there is a lot of color waste in the production of clothes and coloring and so on, but also there is a lot of color waste on objects that they cannot be recyclable because the color is still there. So what about if we are using a color that fades away, and at the end, the final object will be much easier to recycle and even reuse it or recolor it? And also in packaging, that's something that we have been approached by some companies because of the packaging part, because thinking about this idea of a kind of a smart packaging where the fading of the color will give you an idea about for how long this pigment has been placed there, and also will give you, will help you with the recyclability of the packaging itself, and not only packaging, also for like posters and booklets. There are many things that they use a lot of colors and ink, and they have like a super short life. So this thing could be a positive characteristic.
Yeah, I believe so. That it has a positive effect, for sure. Juan, I was just wondering how the engineering students took biology, like the first day. What was their impression? Is it something hard for them to understand? Are they open to understand that this has similarities to engineering, that is structural things? How?
It's funny you ask that, because the majority of them, if I can recall like badly, none of them have done any or biology or chemistry or something
just
like
them,
that.
just general.
Oh, okay.
Even at Elisava, like all your students in general, how they...
Well, okay, I thought we were speaking about the master students. I think the first reaction, it's always like they don't know exactly what you're talking about, and they feel maybe they feel a bit afraid because as they are not familiar with those terms and concepts. But then the immediate next reaction is like a lot of curiosity, because they want to know more on how that relates with the subject, the real subject you're given in class. Imagine yourselves, you're having some mechanics, and mechanics is physics, and for them physics is something like very steady. From Newton, it has evolved very little since. But then you relate that with biology and nature, and they find like a missing link or something like that between the two fields of knowledge. And I think they feel, I don't know, pretty encouraged and happy with that, with the idea of, oh, I'm getting like a novel idea, and I'm going to mix it with some new, or more than a new idea. I'm going to relation in a different way, as I did before that. So I would say it's funny. It's kind of fun for them. Yeah. And for me, of course.
Yeah, I think this idea of design being a transdisciplinary tool to work with, to gain knowledge from other disciplines and bring that knowledge into something that we can approach to, let's say, users, humans, or people, I would say. I think that's really important for any designer that wants to be fit for the future, understand that it's not only design that we need to learn to make things for people or to make design services for people. I was kind of interested in the idea of a glossary for biodesign, because you were mentioning, Laura, before, like biomaterials. And biomaterials is like the word care, that it's been overused and no one else knows what care means. And I think if we could ask any of us in this room, each of us would take a definition for biomaterial. So I want to ask you three what to you would mean biomaterial, which all of them will be right. And also if you could think of any other word that you think, 2 or 3 words, that would be like part of this glossary or future glossary of art, design, and biology, art, design, engineering, and biology. So first of all, the biomaterials. What is to you a biomaterial, Maria? Or anyone who wants to start?
Oh, it's a super difficult question.
It's a difficult question. Well, I cannot start.
It's a question because I'm all the time having this discussion with my students, like, yeah, but this is not a biomaterial. This is, yes, I mean, what doesn't, if it didn't grow in a petri dish, it is not a biomaterial. I mean, everything is material and is made by this by the same building blocks. But yeah. So it's a bit difficult to define what a biomaterial is. Yeah.
But at research level, you would probably link it to a material that is linked to a living organism that has been producing those, right?
I think normally I'm also having the discussion about a material, human-made and non-human-made.
That's an interesting classification.
This is our way also to approaching this biomaterial research world. But definition of what is a biomaterial, maybe Laura.
No, like I said, there's no right definition. That's a plurality of it. Yes. That I think it's what interests us and also.
Well, you can hear me either way, right? Oh. In material science engineering, a biomaterial, it's any material, whether it's natural or not, which has been designed to interact, heal, or replace any biological function. In material science engineering exists. But I think it's a good example, and it's good you ask it, of what Laura was saying about the context. It depends. I think it may vary a lot depending on the context you are. In engineering, for me, and maybe for most engineers, it's very clear, like, no, gold, for example, in your earrings or your piercings, that's a biomaterial. Eye lenses are biomaterials. A graft, any kind of implant. Then we can consider clothing, well, no, because it's not in the body, but, you know, for us it's clear, but maybe it's a good example of context.
Yeah, well, as I said, for us, we are using biomaterials in a general term or as a general term, and we know that it's complex. And here, for example, there is one of the scientists that has been, a biologist that has been collaborating with us, with the artist, that is Anushka Skudi, with whom we have always this debate on what is biomaterials, what you are referring to biomaterials.
She wants to add something.
Yeah, so the thing is that one thing that, for example, for us is pretty, let's say, useful is we are articulating our own glossary. At least to say, hey, within biofriction, when we are referring to transhack feminism, we are referring to this. When we are referring to hybrid practices, we are referring to this. We know that is a huge debate, but we are referring to this. So I would say that within biofriction, even we know that bio, coming from biology, it means that wood could be understood as a biomaterial. We are referring in general terms to those kind of materials, maybe more related to what you were mentioning, and also related to biotech and different, let's say, areas of contemporary biology, and the kind of experimental practices that you can do with them.
Okay, so now we accept that biomaterial is wide and has wide perspectives depending on the context, for sure. Be warned, the students here, maybe I ask you for definitions of biomaterials too. So, any questions so far? Anyone? No? Yes?
Oh, really? Oh, I know.
You might have to come here.
Oh, of course. Of course.
My question is to Maria. Have you thought about the carbon footprint of the microalgae pigment and like the scalability and industrialization of it? I mean, I just want to know the kind of energy that's required to produce it, and then et cetera, et cetera.
I didn't hear the question.
The carbon footprint of the microalgae pigment and, you know, the scalability and industrialization of it. Have you thought about it? And if you have, like, what is the process?
microalgae are already growing on a big scale, but it is true that that depending on when you are growing algae can become an expensive product. That's why we wanted to work with waste. And yeah, ideally, I will be doing this project in Spain, where the energy needed is much lower, because you can do this process of bioremediation outdoors to have your waste of your company out of your company. With the sunlight, you just can produce this biomaterial, this biomass, while cleaning the water. But yes, I mean, in the project that we did with the wastewater, we checked the efficiency, and it was positive because everything in the system was gone. But it is true. I mean, everyone who is working on microalgae, doesn't matter if it's for pigment or for food or for biodiesel, is dealing with aspects of how it's called efficiency. But yeah,
Oh, okay.
I don't
Thanks.
know if I answered your question.
No, that's fine.
Okay, we have a question from the audience, and don't worry that I don't forget about the glossary, so I'll still ask you some of the words. But we have a question from the audience here. How would it be possible, that's for you, Laura, well, how would it be possible to collaborate with Biofriction Lab in Barcelona? Do you have residencies, programs, academic funded research projects? It's Emma Sicher.
Okay, the thing is that Biofriction is a 2 years project and is about to end in September. So, as I said, it's a project based on transnational mobility that it's been pretty difficult within the pandemic, but it has different, let's say, activities like residencies, workshops, and we will be doing a summer lab in July here in Barcelona, so stay tuned. And I said that the wet lab is the node of Biofriction here in Barcelona, and in the wet lab there is a collective in residence that it's not, let's say, it's not related directly to Biofriction, even we are collaborating all the time, and they are, you know, like Biofriction, it has this kind of different nodes, like the institutional one with the capital B, and all the projects, artists, and networks that are the condition of possibilities of Biofriction, pre-Biofriction, during Biofriction, and for sure post-Biofriction. So I would say that please join us in the wet lab. In the wet lab, yes, why not? In the summer lab.
Okay, so we have summer lab, Biofriction summer lab. So this summer, I guess.
Yes.
Great. So just a few words. I just want to add to this, like at the beginning of this glossary, and bioremediation. I think that's a really interesting word. Also, Juan, which one would you pick from your presentation? I'm not making you think. What kind of word would you pick from your presentation that explains, or that can build up a glossary, or start building up a glossary?
Probably bioregeneration.
Bioregeneration. So bioremediation, bioregeneration.
Just one, you said, right?
No, I said more, but we can start with one by each, and that's fine. I mean, this is a reflection on how this transdiscipline needs some kind of structure or glossary, some kind of vocabularies that we train our students or future professionals with the right wording, understanding the context, and when we talk about this world of different hybrid disciplines. That's it. So bioremediation, bioregeneration. And if you have one, Laura?
Yeah, I have one. It's not mine, of course, but I would say that it's important, at least would be ideal to have at least one term referring to ethics, and I'm not going to use bioethics, but for example, speculative ethics, no? Because the kind of practices that you are developing as a designers are giving us speculative tools in order to also to think of different articulations of ethics that maybe are not deontological anymore, are not based just on duty. So I would say that this speculative potential is giving us a lot of materials in order to try to articulate different ethical approaches. And just if it's interesting to you, there's a book by Maria Puig de la Bella Casa that works on this. It's called Matters of Cares, and it works on the notion of speculative ethics.
Thank you. Well, at least we have strong words to start with the glossary. Okay, I invite you to do your own, to start building this. Maybe we can do it collaboratively. And I think there's no more questions on the internet. And if there are no more questions here in the audience, I think we can close the debate. We can close this great, inspiring lecture with these three speakers. We hope that this opens up debates, experimentations, projects, collaborations, and to start building up this, or growing this transdiscipline of art, design, biology, and engineering. So thanks everyone for being here, also those who were online. And that's it. Well, before closing, thank you, Angar, thank you, Ludovica, and all the team here. It's been great managing of this lecture. So thanks, everyone, and that's it. Thank you.